Here, Paige, this is what you're missing.
Good morning, everyone. Welcome to our meeting today. Thanks everybody for being here as we count down towards spring. That's how I'm looking at it, with a foot of snow on the ground. We'll soon have good weather around the corner, and hopefully be able to get back to real meetings again in the near future with the vaccine rolling out and warmer weather coming. But thanks for continuing to be here on these weekly Zoom meetings. It is important that we keep our club alive until then, and I think we're really close now. This morning, our invocation is going to be led by John Williamson, and then Dave Williamson will lead our pledge. So whenever you're ready, John.
Okay, let's pray. Father, we thank you for those whose work sustains our nation and this community in which we live, for all who create the wealth by which we trade, for those who grow and provide our food, or who in industry, commerce, and transport bring it to our homes. We thank you for those who day and night maintain the public services, for the police, for those who respond to emergencies, and for all whose work in health and healing and social care. Teach us to remember that all our lives depend upon the work of many minds and hands, and we pray that we may live thankfully and in unity as members of one human family. All these things we pray in your name. Amen.
Amen.
Good morning. Please rise and join me in the Pledge of Allegiance. I pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Thank you, John and Dave. Do we have any guests to welcome this morning? If you have a guest, or if you are a guest, please unmute yourself and make an introduction.
Hey, Dave, Lyle Brown is on again, and I'm happy to say that he is going to be putting his application to join the club. So I want to say welcome to Lyle.
All right, welcome, Lyle.
Thank you. Thank you very much.
Excited to have you join us.
Absolutely.
I'm excited to join the club.
Thanks for being here again today and for your interest in membership. And I think, like I was saying earlier, we're going to get back to being a real in-person meeting club before too long. So thanks for hanging in there with us. Other guests? Right, how about announcements this morning? If you have an announcement, please unmute yourself.
I have an announcement. I sent out an email yesterday, but wanted to let folks know that we have some commemorative jerseys from the polar bear outing. And if anybody would like to buy one, $15, just let me know, send me an email. Thanks.
Thank you, Susan. Other announcements this morning? Wow, quiet group today. Okay. Well, if we don't have any announcements, then I am going to ask Susan West to move right into introducing our speaker for the morning.
All right, fantastic. Well, guys, I am so excited to introduce to you Elaine Mardis. Dr. Mardis, had the opportunity to meet Elaine this past summer, and we got to talking about what she does at Children's Hospital. And I just thought the idea was simply, I don't know, I told her we're all a bunch of inquisitive people, and the genomic research going on right now, I think, is very interesting. Elaine is the co-executive director of the Institute of Genomic Medicine at Children's Hospital, and she's been at Children's since 2016. She started her education at University of Oklahoma in zoology, and then got a PhD in chemistry and biochemistry. And then she did her postgraduate work, sorry about my dog, at Biorad Labs. And basically, the research she has done is amazing. She's written 380 articles in journals. And anyway, I think I'll turn it over to her so you don't have to listen to my golden retrievers. I'll hand it over, Elaine.
Thanks so much, Susan. Good morning, everyone. It's really a pleasure to be here today and to represent the work that we're doing at Nationwide Children's Hospital. So I am a scientist, as Susan mentioned. Scientists tend to speak from slides, and so that's what I'll do this morning, just to walk you through some background material, sort of focusing on adult oncology first. And then use that as a springboard to describe the program that we've developed out in pediatric cancer at Nationwide Children's Hospital over the past 3 years. So I'm going to share my screen now and get us going. And I'm pretty flexible about interruptions. So if there's something that just doesn't ring true or you're not clearly understanding what I'm talking about, please feel free to unmute, speak up, and let me know if I can explain something better. So I just entitled this Pediatric Cancer Precision Medicine and the Rasmussen Institute for Genomic Medicine at Nationwide Children's Hospital. And as Susan mentioned, the Rasmussen Institute was opened in 2016 when I and my longtime research partner, Rick Wilson, were recruited here from Washington University in St. Louis. And our real mission at IGM is to introduce genomics, which I'll talk about today, into the practice of medicine, cancer being one of the areas that we focused on initially, and I'll talk about some new areas of focus right at the end of the presentation. So we've known for some time now that cancer is related to the genome. If you look at a book that examines the history of cancer, like The Emperor of All Maladies, that was written a few years ago and won the Pulitzer Prize, we know that cancer was first described in writing by the ancient Egyptian papyri that were attributed to the physician Imhotep. And that was around 4,600 years ago. Certainly our knowledge has advanced since then, and I'll talk about that today. But in particular, I think, advances in science are all about visionaries. And certainly this German zoologist, Theodore Boveri, was a visionary in that way before, we had some of the tools that I'll tell you about today to decode the genome, he recognized that there was a genetic basis for cancer, and that was in the early 1900s. And he made this hypothesis after he was observing chromosomes under the microscope. Moving forward in time, in the early 1970s, one of my scientific heroes, Dr. Janet Rowley, shown here on the left at her microscope, began looking at leukemia cell chromosomes. She was a technology adventurist, and so she developed methods to condense chromosomes, much like the picture on the right-hand side here from her work. And she began examining chromosomes from patients with the same type of leukemia and was able to come up with these very common chromosomal changes. What's demonstrated in the right are essentially a chromosomal fusion, two pieces of two chromosomes that aren't supposed to be put together, actually coming together. And this was diagnostic for a specific type of leukemia called acute promyelocytic leukemia. This chromosomal translocation or fusion actually brought together two halves of two different genes, which resulted in a completely novel protein. And that protein is a driver for this type of leukemia, causes it to occur, and is now the focus of targeted therapies, which are very successful at curing patients with acute promyelocytic leukemia. So it's this sort of recognition of the change in the genome, the development of a therapy to address or shut down that cancer driver that's resulting from that genomic change that I'll talk about throughout the 20 minutes or so that I have with you today. I want to do a little bit of background first in terms of terminology. One of the things you might be wondering is, what is a genome exactly? And so here is a simple definition. If we look inside each of our cells, in the nucleus, we find a set of 23 chromosomes. And this is quite simply our genome. This is the instruction set for making a large complex organism like a human being that lies in the DNA. And you would think that DNA then by inference would be a very complex molecule. It is in some ways, but excuse me, the basic building blocks of DNA are four. They are chemical building blocks referred to by the single letters A, C, G, and T, which stand for larger names for these building blocks. And the chromosomes themselves are actually quite large. So if we unwound all of these, they would be about 6 feet in actual length. So obviously a human cell is way too small to see with the naked eye. You need a microscope. So there's a systematic way to condense these chromosomes by winding them around proteins called histones to condense them into what would fit into a cell nucleus. And the human genome, which I'll talk about in just a minute, is quite large. So in addition to the 20,000 genes or so in our genome, which is about 2% of the total size of the genome, 3 billion base pairs. And so we are a quite complex organism. It might surprise you to know that very simple organisms like fruit flies that you might see at the supermarket or roundworms that live in the soil also have about 20,000 genes. So before you feel bad about that, it's just that we have, as human beings, many ways to express these genes at different times, to make modified copies of those genes, resulting in different proteins. And this really, in essence, is why we are such complex organisms compared to a worm or a fruit fly. So if we want to talk about the genome for a minute, one of the things that we recognized early on, even before we decoded the human genome, is that we wanted to learn more about this connection between cancer and the genome. We really had to study and understand what was in the genome. And this, again, visionary, Renato Del Becco, who's a Nobel Prize winner, in the mid-1980s, even before we had sequenced the human genome, basically said, look, if we want to understand cancer, we have to decode the genome, and we have to either do this gene by gene, or we have to sequence the whole thing at once. And in 1986, that was a pretty big, audacious goal that was basically not attainable. But his comments and thought leaders like Jim Watson, for example, who won the Nobel Prize for discovering the structure of DNA with Francis Crick, and many others began to actually think about what it would take to sequence the human genome. That, combined with the interest of governments worldwide, really resulted in the sequencing of the human genome. This is a project that I was very proud to be a part of during my time at Washington University, where our laboratory, one of the 3 large genome centers in the United States, supported by the federal government, produced about 20% of the total human genome, that 3 billion base pairs that I mentioned. And we then went on to to refine the sequence of many of the major human chromosomes, some of which have known cancer genes, including chromosome 7. So by 2003, we're leaping forward now in time by a few years, we had finished the human genome reference, as it's referred to now, this human genome template that helped us to understand where all of the genes were located and gave us really the ability to start exploring in cancer samples the possibility of finding the mutations that actually lead to cancer onset. So that was 2003. We didn't really waste much time moving that forward. So you can see here a paper that we were part of in 2004. What we actually started with was to look at not just cancer, but actually response to new cancer therapies. So in this study, which involved only about 19 patients at the time, we had 2 distinct sets of patients. These were all lung cancer patients. They were being treated with a very new class of drugs called TKIs, or tyrosine kinase inhibitors, a so-called targeted therapy. And some of these patients were having dramatic responses, wherein having, advanced lung cancer with metastatic disease, and taking these drugs, they were actually having their cancers literally melt away in only a few weeks to months of being on the therapy. So those were the responders. And by contrast, there were a subset of patients who continued to have advanced disease and no response. So this raises an immediate scientific question, which is, why do some people respond and some people don't? And what we did in this study was to just focus on specific genes. The targets of these tyrosine kinase inhibitors weren't exactly known, but there were suspect genes that were likely to be involved. And what's shown here is a gene referred to by an acronym, EGFR. I'll tell you what it means, epidermal growth factor receptor. This is a protein that sits at the outside of our cells. And in cancer patients, as we're showing here, that have responses to these drugs, there were specific mutations in this domain of the protein, this part of the protein that is called the tyrosine kinase. So it's really the active part of EGFR as a natural protein. In cancers, it gets mutated in some patients, especially in lung cancers. And what we were able to make the connection with here is people who had these mutations were the responders to these drugs. And that was true in about 80% of the patients. So this is really one of the very first examples of understanding drivers, these mutations in specific genes that led to the onset of cancer, and linking in the response to a targeted therapy to the presence of these driver mutations. And I'll show you how this is now developed out since 2004 in just a moment, because we've learned so much about lung cancers from large-scale sequencing projects. First, want to just take a small segue because I'm a technology geek by training. And so what I have worked with throughout my whole career is technology in the context of genomics. And what I'll tell you about in terms of this revolution in genomics that's been going on since about the early 2000s is that the technology has really been the underlying driver in our ability to not just focus on single genes, like in the study that I just showed you, using instruments like this instrument here on the left, which was really back in the day, the state of the art, and what we use to sequence the human genome. But the ability to look at the entire genome in an unbiased way, not trying to guess which genes are involved in cancer like we did in the previous study, but just letting the genome tell us what's been mutated, what's been altered, really was reliant on the development of new technology. So these just look like big plastic boxes to you, I'm sure. But if you look at the bottom, you can see that on the left, the capillary technology that we use to sequence the Human Genome Project was relatively slow and expensive. So the Human Genome Project was about $15 million to just sequence one genome if you went back and did it today with this instrument. And for the Genome Project, it took us about 5 years and was involving many groups across the world. By contrast, the NovaSeq instrument, which you see on the right, available from a company in California called Illumina, literally can sequence about 24 whole human genomes in a couple of days. And the cost there per genome is not $15 million, it's $1,000. So that's a pretty dramatic change in a relatively short period of time. And of course, the instrument that I'm showing you here is just the newest, greatest instrument. These sequencers from Illumina have been in our lab since about 2006, with a variety of technology innovations over time that lead to the single instrument throughput that you're seeing here. I also like to euphemistically refer to the NovaSeq as the $1 million washing machine because, let's face it, it looks like a washing machine, but it costs $1 million. So it's a little bit different, I guess, than a traditional washing machine. So what can we do with this new technology? Well, very early on, our group was able to show that in an unbiased way, we could take these next generation sequencing instruments, isolate DNA from cancer, cells from a patient, isolate normal DNA from unrelated tissue. In this case, we're talking about a patient with leukemia, and a skin punch that's usually taken when the marrow is extracted from the hip bone of leukemia patients, a so-called marrow biopsy. And we were able to sequence both of these genomes with this technology, align those reads, the sequencing data, if you will, from these two related tissues from a single individual, and then compare the mutations that we identified in the tumor to the normal. In other words, what's changed about this patient's genome that wasn't there at birth, their normal genome, and is now unique to the cancer? And with this audacious experiment back in the day, which was way too expensive, you can see that it took us about 3/4 of a year just to generate the data. And the cost of generating the data was not $1,000 like the instrument that I just showed you, but actually cost us about $1 million to complete. This was from private philanthropy because the government, NIH, basically said, no way, it's too expensive. The computational analysis of the data doesn't exist. It's way too early to do this experiment. So we were able to engage a private funding individual in St. Louis at that time to donate $1 million to do this experiment. What we identified in this comparison of tumor to normal was we found 10 genes that had mutations. 2 of these were previously known genes from AML studies, so we felt confident in that. But we didn't know whether 10 mutations was too many or not enough, et cetera. I can tell you that several years later, having used these same approaches and sequenced through over 200 patients with AML, we identified that the average number of mutations per genome was about 12. So we weren't too far off of the mark. But importantly, doing this experiment at the time that we did it really set up the paradigm for using next generation sequencing to look across the genome in an unbiased manner, not to have to guess, to look under the lamppost, but actually to just let the genomes tell us about what the changes were that were unique to the cancer. And this type of study then was an inspiration for worldwide sequencing of cancer genomes. And this is really, I'm showing here, the full thrust of this approach, which has been rolling out over the past 10 years or so, and is now complete. We know a lot about adult cancers in particular. I'll get to kids in just a minute. And specifically in the United States, the Cancer Genome Atlas, or TCGA, as it's referred to, shown here, was essentially a very large science project that involved sequencing through tens of thousands of cancers of different types. You can see some of them listed here. And really looking at multi-omic characterization. So not just sequencing the DNA, but also looking at RNA, which is the intermediate between DNA and proteins. Proteins are the actual working horse of the cell, but also looking at modifications to DNA like methylation, et cetera. So what I'm trying to represent with this slide is the compendium of knowledge that's really been accumulated again over the past 10 to 12 years that is now fueling this revolution in the way that patients are diagnosed with cancer and the way that they're treated. And this is just a flow diagram that illustrates what I just said from a review that I wrote a couple of years ago, where the real, idea behind writing this review is how genomics has now started to impact the clinical care of cancer patients. And I won't bother to go through the details here for sake of time, but just suffice it to say that by sequencing the cancer DNA and sequencing the normal DNA, germline is what we call it scientifically, we can actually identify sets of mutations that are highly informative for each and every cancer patient. I'll try to illustrate that in a moment. The other aspect that's really been important in this whole journey is the interest of drug developers, including pharmaceutical companies, to marry together what we're discovering that drives cancer in the human with drugs that can actively shut it down, much like these early tyrosine kinase inhibitors that I was telling you about a minute ago in lung cancer. And this graphic here shows that we've been incredibly successful. This is just a figure from our annual progress report from the AACR. I'm the former president of the American Association for Cancer Research. And we put out a report every year that sort of marks the progress in cancer. And this just illustrates how far we've come in that regard just in the last year from 2019 to 2020. So 16 of these so-called molecularly targeted therapies, much like the tyrosine kinases I was telling you about, and additional applications or uses for drugs, 15 just in the last year, where they've been approved in one cancer type and now are approved in treating a different cancer type. So I'll just sum up what I've said in terms of background and then switch to pediatric cancers here. So let's look at lung cancer for a minute in terms of the transformation over time. If you look in the far left pie graph here, you see how in a pathology sense, historically, this is how lung cancer has been diagnosed, where looking through the pathologist's microscope, doing a variety of stains, one could basically say, okay, this is either an adenocarcinoma, a large cell cancer, or a squamous cell cancer. And that's about the level of precision that could be applied to diagnosis in, in the era from the 1950s to the early 2000s. The use of cancer genomics to decode non-small cell lung cancer, which is the 70%, also called lung adenocarcinoma, is shown in that second pie graph, which is more to the right. And what I'm showing here is that by genomics, we've been able to identify these 3 or 4 letter designations around the pie graph, different genes that are mutated and drive the onset of that cancer. So you can see in the left-hand side, the EGFR that I mentioned in that early study that we produced coming out of the human genome. So that's about 15% of lung cancers. And there are other genes that are listed that are known drivers. And of course, you'll notice immediately that about 42% of lung cancers still don't conform to a specific diagnosis. So we don't know everything, but we know a lot. And let's show how this, and along with the development of new drugs, has really benefited patients with cancer. And this is really the essence of precision medicine, that term that I used earlier. So current day, let's say a patient goes in, hasn't been feeling well, having some other associated health-related issues. There would be an imaging done. A mass might be detected in the lung, and that is typically biopsied in a procedure called interventional radiology, where you image the mass and poke it with a needle to pull out some tissue and evaluate that tissue. We still evaluate that under the pathologist's microscope, but part of the tissue goes for next generation sequencing-based analysis, much of what I've told you about already using these advanced sequencing instruments. If we find, once again, an EGFR mutation is present, there are now a multitude of drugs, they're listed here, that are available to treat this patient. And this is now not for metastatic patients, but actually the first line of therapy if these mutations are identified. EGFR can combine with one of these other driver genes called KRAS. You'll see it in the pie graph at about 30%. These are often mutations found in smokers or former smokers. We also know that because of the smoking, there's an elevated mutation rate because smoke has carcinogens in it that cause mutations in DNA. And so the elevated mutation rate in these patients now allows you to get in the first line, again, new types of therapies called immunotherapies. You may have seen commercials for these on television recently. And immunotherapies have revolutionized cancer therapy in many, many ways. They led to the 2018 Nobel Prize to Jim Allison and Toshi Honjo. And so immunotherapies are now being used to treat patients with these high mutation low tumors. And then lastly, there are genes that get fused together, much like the fusion that I told you about in the context of Janet Rowley's work in leukemias. These also happen in solid tumors and in the case of ALK fusions, yet another gene that's altered in lung cancer, there are targeted therapies that can be very effective for these patients. So this is really the essence of precision medicine. Test the tumor for drivers, identify a therapy, if available, that will address these drivers, and in many ways alleviate the cancer, sometimes for very long duration. So the essence of this, I like analogies, is that this is new technology, but a much clearer picture, a much more precise way of diagnosing cancer. So you could think about back in the day, if you didn't feel well, you went into your doctor, you get a chest x-ray, and you can kind of see this hazy blob that is the cancer for this patient using x-ray, which is really best suited for looking at solid tissue like bones. But a CT scan shows that cancer quite clearly. And so really the CT scan is our version of cancer diagnosis using genomics. So I'll just spend the last few minutes talking about our work now that I've set the table for adult oncology in terms of how we really want to bring this precision medicine approach to kids with cancer. And the compelling reason here is that cancer is the leading cause of death by disease in the developed world in children over 1 year of age. And in particular, my research interest, which lies in brain cancers have now surpassed leukemia as the leading cause of cancer-related death in children. So why are pediatric cancers different? Well, what I haven't told you is that 5 years at WashU was spent working with St. Jude to do a similar large-scale evaluation of pediatric cancers, much like the adult cancer TCGA project that I told you about earlier. What did we learn from that? Well, we learned a lot of things, and these are all the reasons why pediatric cancers are difficult. So first of all, adults typically have a large number of mutations that we can identify, but pediatric cancers, much like the AMLs I told you about, typically actually have a very low mutational burden. So not many of these obvious druggable targets come out of just looking at DNA, much like I showed you a minute ago for lung cancer. That means we have to look in other places like sequencing RNA, for example, or looking at proteins to really find druggable targets. The other challenge is that the FDA and clinical trials in particular have really been focused on adult cancers. Pediatric cancer is still a rare disease. And so in some cases, it's hard to come up with enough children, even across the United States, in a very large trial open at many hospitals to really satisfy the FDA requirements for approval of these drugs. And so we're often quite far behind in terms of using FDA approved new targeted therapies or immunotherapies in pediatric patients. But it is possible, and I'll show you an example of that in just a moment. But in particular, we're just far behind in understanding the cancer biology, as well as the interactions with the immune system that have led to some of these revolutionary new therapies. However, we're also far behind because our government really hasn't taken a step forward until very recently to try to promote this basic approach to precision medicine in the pediatric setting. And in some ways, we're being eclipsed by other governments that have taken this, sort of initiative. So I'll show you a couple of examples. The furthest along are the Germans with their KITS Inform trial that's been studying pediatric cancers in the clinical setting since 2015. Also, the Aussies have put together a trans-Australia, 8 different pediatric hospitals, most recently reporting about 400 patients that have been studied through this protocol for enrollment onto treatment with targeted therapies. And then just to our north, a little more recently, the Profile Precision Medicine platform was announced involving these 3 centers, Vancouver, Toronto, and Montreal, but again, collecting patients from across Canada for study using these precision medicine genomic profiling initiatives. Importantly, as part of the Trump administration, there was an announcement halfway through to target funds for a childhood cancer data initiative. This is an important step forward that brings in data from data producers like ourselves and other children's hospitals to try and increase the knowledge around childhood cancer. But we really need a molecular profiling initiative to go along with this, and we're hopeful that that will happen in the Biden administration. So what is the other aspect of pediatric cancer that I'm not telling you about? Well, it's the survivorship aspect. You may have heard this term, and you may know cancer survivors. The challenge here is that we've done really well, especially in the adult realm, treating patients with cancer, and to some extent in pediatric cancers as well. I told you that leukemia is now second to brain cancer, and obviously we've been very successful in treating patients with leukemia so that it doesn't come back. And we have an increasing number of cancer survivors in the United States. But again, children really are falling behind in terms of the aspects of cancer survivorship that we don't like to acknowledge. And that is that by treating children with chemotherapy or radiotherapy, radiation therapy, or the combination of the two, we have still a significant amount of post-therapy challenges for these children. So this is just a recent paper from the Journal of the American Medical Association's oncology-focused journal, looking backwards at kids that were diagnosed in the 70s, 80s, and 90s, and showing that while some progress has been made, adult survivors of pediatric cancer still remain at risk because of the side effects of chemotherapy and radiation therapy for a shortened lifespan. And this includes a significantly elevated rate of secondary cancer occurrences. This would go away in an era where we use targeted therapies in kids as well as immunotherapies. And so our studies at the Institute for Genomic Medicine are really committed to changing this reality. Now, I won't walk you through all the nuts and bolts of how we do what we do. I'm just going to show you a simple example of a child that we were able to help to sort of set the stage for how this is all working. So in our protocol, which has been open since January of 2018, we've been able to study over 300 children. I'll show you the aggregate of that in just a moment. Let's just focus in on this child diagnosed in 2013. You can see on the far left the fact that there's an MRI of this child's brain, and the white spot that's on the far left-hand side of this image is a cancer. This is not a brain cancer per se. It's actually in the meninges, one of those membranes that surrounds the brain, and it's a sarcoma as designated once it was removed by surgery. You can see gross total resection above the gold bar. And then the postoperative imaging in 2014 shows that it's no longer there. This patient then underwent proton therapy, but recurred in 2018. The big red arrow points to the recurrence. This recurrent cancer was also removed by surgery and studied on the IgM cancer protocol because this patient now has a recurrent cancer and may be available to certain therapies that would be in the setting of precision medicine. So we studied this patient's DNA from the tumor and DNA from the blood for the comparison normal, much like that early AML patient that I told you about. So we always compare tumor to normal. And what we identified were two things. One, that there is this mutation, it's shown on the left-hand side here, in terms of in the germline blood, the normal. This is inherited from the father, we know, and the father actually had his own problems with cancer in terms of colon cancer diagnosis. And it's also present in the tumor, which is what we call the somatic tissue. But from the standpoint of a precision medicine target, we were able to show that unique to the tumor, shown down here on this lower right-hand panel, that there's another mutation. This is in a gene with a long acronym and a longer name called platelet-derived growth factor beta. And this is a cancer-involved gene. The protein structure of PDGFR beta is shown on the far right-hand side of the slide. And in particular, this patient has a mutation in this part of the protein called the juxtamembrane segment. That's the part that spans the membrane. And what we know about PDGFRB is that it has targeted therapies that will address this activating mutation or gain of function mutation, which we think is driving this patient's sarcoma return. We also looked in RNA, that intermediate molecule that translates the DNA information into proteins. And you can see on this graphic, which is comparing our patient, CH21, to all of the other patients that we had sequenced that so far, just for the expression of this gene in RNA, that the expression level is quite high. This is another indication that this is a driver gene. And then finally, we do what's called a pathway analysis. So if the gene is overexpressed, is the pathway that it participates in also activated by that overexpression? And hopefully you can see here that PDGFR beta is this sort of red-pink triangle. And the fact that many of the genes that are listed out here around this diagram related to the PDGFR beta pathway are also red, means that this is an activated pathway. So if there's a way to drug this pathway, then we can effectively shut it down and turn off the motivation for those cancer cells to grow. They will then die, and the patient will have relief from their tumor burden. And this is in fact the case. This is just a complicated diagram showing the signaling pathway that we're focused on here, and indicating that if we address this membrane-bound protein that's mutated, PDGFR beta, with one of these drugs, we can actively potentially shut it down. And because this patient has failed the standard of care, the first proton therapy and surgery intermediate, they're eligible for this one of these drugs to be paid for by insurance to test the response in this patient. So I'll just finish with this example by showing you that on the far right-hand side of the slide, we have a recent scan of this patient's brain. And you can see that as of January of this year, he remains with what we call NED, no evidence of disease. Continuing to take the dasatinib therapy that was identified by his oncologist at Nationwide Children's Hospital, and living a healthy, happy life as now a 10-year-old. So this is exemplary of what we would like to do. Is every case this simple? No, it's not. But what you can see in aggregate at this point in time is that we're having a significant impact on our cancer patient population at Nationwide Children's Hospital. So we've now evaluated over 250 pediatric cancer patients across brain cancers, sarcomas, and a few in the hematologic blood cancer space. Over 90% of these patients have at least one what we call medically meaningful result that helps our oncology team identify best therapeutic approaches. So this is a very high diagnostic yield for this complex series of tests that we're looking at each patient with. And as I've already mentioned, these results are most impactful in patients that either have rare cancers for which there may not be a standard of care identified or targeted therapies, or where the cancers have returned, much like that child that I just showed you. And the standard of care which exists has actually failed, and so they're looking now for new options. And we can also help to refine the diagnosis when it's not clear from the pathologist's microscope what the diagnosis is, and in turn, knowing the diagnosis can help in terms of identifying the right therapy or the therapeutic approach for these patients. So I'll just tell you in finishing that what I'm missing out on this morning, because I'm happy to be here talking to you, is that on Friday mornings, we actively are reviewing patients in our brain cancer cohort in the clinic, and returning these kinds of results in the context of looking at the radiology imaging, talking to the oncologists and pathologists about what we found, and promoting forward the ability to really use this precision medicine approach to help determine what their treatment will be moving forward, if that is indeed indicated. And in essence, in that regard, we are fulfilling our goal or mission at IGM, which is to introduce the use of next generation sequencing and analyses into the practice of medicine. So that's been a really great journey to happen over the last 3 years, but this is not where we're going to stop. So I just want to finish today by saying what's next, and I won't talk you through all of the many boxes on this slide. You can read them for yourself. But I think the bottom line is that genomics, what we are practicing at IGM, has and will continue to have significant impacts on our pediatric patient population across multiple different indications. This includes babies in the neonatal intensive care unit, for example, children with epilepsy, and into the future, children with different infectious diseases or immune problems, just to name a few that are on here. So this is really transformational. We're really excited to be a part of such a vibrant hospital system that is really embracing these new modes of diagnosis. And we're especially thankful for a lot of the philanthropic gifts that have supported the work that we've done so far, most notably the Nationwide Insurance Foundation Innovation Fund, which has steadily contributed on the order of about $10 million per year over the last 4 years, so that we can do the kind of work that I told you about today. So I'll just finish by saying thank you for your attention, and certainly happy to answer any questions that you might have about the work that we're doing.
I have a question.
Okay.
You seem to do a lot of work on lung cancer and stuff like that. Have you ever done any work with colon cancer?
Oh, sure. So colon cancer is actually a really important area for young adults, which is where we're focusing a lot of the work that we're now planning to do with OSU. And in particular, colon cancer is in increasing prevalence in young adults. And we don't really understand all the reasons why. So there will be a new program that's being announced as part of the OSU Comprehensive Cancer Center, where I also am in the leadership group with Dr. Pollock and others, very much focusing on what we call, it seems like everything's an acronym nowadays, AYA, so adolescent young adult cancers, applying these exact same genomic assays that I told you about today to those studies and really trying to understand better how to treat those patients.
And have you also studied some of the work of Henry Lynch, also?
Oh, yes.
Lynch syndrome?
Indeed, I have. In fact, I had the pleasure of meeting him once. He spent most of his career at the University of Nebraska, and I'm a native Nebraskan, so it's a bit of a point of pride. We do have a test that we introduced for in collaboration with Paul Goodfellow at OSU, who is a GYN oncologist there, that tests women with endometrial cancer, because Lynch syndrome, which was defined by Henry in his seminal work throughout the years, is one of the main contributors to the development of endometrial cancer, as well as colon cancer. So it lends itself to susceptibility to multiple types of cancer. And we now have a test that we have offered out across the state of Ohio, where the provider, the GYN oncologist who's diagnosing these women with endometrial cancer, can send in a sample of the cancer, and we can do the testing and determine whether that patient actually has Lynch syndrome or not. And of course, that can be very impactful, not only for the patient, because there are new drugs now that are approved, these immunotherapies that I mentioned, that can be used to treat these patients directly, but it can also inform any of their siblings or children about their possibility of also having susceptibility to cancer. And they can go ahead and be tested as well. One of the things we know about understanding susceptibility to cancer is it changes how your medical treatment occurs, so that those cancers can be identified earlier and maybe more successfully treated, as opposed to being found late and having a very poor outcome, which has been the typical case in the past.
I've been tested, and I'm a Lynch syndrome patient, and my children have been tested and they are not. So
Yes, and
I admire your work. So
thank you. And that's really the reason for doing it is, as I'm frequently fond of saying, knowledge is power, right? And so by understanding your susceptibility, hopefully you either understand that you don't have to worry, or you understand that your life in terms of medical care will change over time, but that's a good thing. So thank you for mentioning that.
Doctor, I have a quick question. I know we're short of time. It seems like St. Jude's gets all of the press for the children's cancer work and research, and they have an incredible endowment philanthropy program.
Yes.
Is the same, does children's work with St. Jude's in terms of should we be locally gifting and thinking about supporting children's in that philanthropy space for the same kind of research?
So it's a really great question. It is true that St. Jude has a very high profile. They're the Sons of Lebanon, which is sort of their philanthropic arm, if you will, is extraordinary. They have over $1 billion a year that they attract in philanthropy that really drives the engine that is St. Jude. And wonderful people there. We really enjoyed working with them on the pediatric cancer genome project that I mentioned briefly. I will tell you that while St. Jude is a remarkable research hospital, they actually don't treat that many patients with cancer. So I think that's the sort of differential, if you will. We have a lot more beds and treat a lot more patients every year at Nationwide Children's Hospital with cancer than are treated at St. Jude. I do know that recently they've built or are building a new hospital tower at St. Jude to increase the number of beds, but it's actually a fairly small bed number of hospital. So the answer to your question is, yes, of course. I'm sure Nationwide's foundation would love to hear from you. Certainly these philanthropic gifts that I mentioned from companies are also helping in this regard, but certainly individuals can be transformational in that regard as well. And I guess the other thing that I really wanted to say is that this is not so much just about our hospital either. Pediatric cancer is a rare disease, so we need to share data, and we do actively share data, not just within other hospitals in the US, but actually out to international entities like some of the ones that I told you about, Australia, Germany, et cetera. Really, by sharing data, we can transform these rare diseases that are pediatric cancers, because we learn from other people's data much like they learn from us. So when it comes to St. Jude, we actively are sharing data with St. Jude and they with us. And this enables us to move our ability to diagnose cancers to the next level, in particular using advanced approaches that you may be hearing about in the press, like machine learning or artificial intelligence. Those are real things, and they really require large data sets to operate on. So I don't know if that exactly answers your question, but we don't want to just sort of stay where we are in terms of just looking at cancer patients from Nationwide Children's Hospital. You may have also read that Nationwide has some outreach. We've recently partnered with Dayton Children's Hospital. We've recently had a major stake in Toledo Children's Hospital. We have strategic partnerships with Cincinnati and Health Rainbow Babies in Cleveland. We would really like to start making this initially an Ohio-wide capability to study patients like this, to work with the oncologists at those hospitals that are treating the patients and understand the data that we're generating and how it can transform the care of these children, and then take it to the next level. Doing that will require this nationwide initiative that I mentioned, which hasn't happened yet, but we're quite hopeful that it will.
Great work. Thank you.
Thanks.
Okay, I'm gonna stop the questions now just because we're out of time. Dr. Elaine, thank you very much for your
Thank you.
presentation today and for the important work you are doing to extend and improve the lives of children who have cancer, and helping their family. Thanks for being here. Let's give a hand to Dr. Elaine.
Thank you. Really appreciate it. And all the great questions.
Next week, we will hear from Jenny Stotts and Laurie Reinbolt about Rotary membership. So be here next week, same time, same place.
Thank you, Doctor.
Everybody have a great weekend.
Thanks again.
Bye-bye.